Welcome to Chapter 17, Biotechnology and Genomics. This will be the wrap-up of our genetics section of the class and our wrap-up of the class in general, so it's exciting. So let's start with an introduction to biotechnology. Biotechnology uses biological agents for technological advancements. Mostly this applies to medicine and agriculture, things like vaccines, antibiotics, genetic modifications to increase crop yield, fermentation, treating oil spills, producing biofuels. I mean, the examples really just go on. Scientists use some basic techniques to manipulate genetic information, and we used quite a bit of these this semester in lab. So DNA and RNA extraction, gel electrophoresis, nucleic acid amplification by PCR polymerase chain reaction, hybridization, southern blotting, and northern blotting. The first step to being able to manipulate DNA or RNA is to extract it from the individual. So it has to be isolated from the cells. A lysis buffer will break down the cell. Proteases can be used to break down proteins to access the DNA. Ribonucleases break down RNA. So we did that part, and then we moved on to gel electrophoresis. Because nucleic acids are negatively charged, they can be mobilized by electric fields. So putting it in that gel and running the electric current across it, we were able to separate molecules based on size. And then you can look at the gel to see the different levels of, you see the ladder on either side here and the different molecules in the middle. And you can look at that under UV light. Nucleic acid fragmentation amplification by PCR. So this uses specific DNA regions. It amplifies them for analysis. So it finds the best section of DNA, the purest segment, the most concentrated segment, and amplifies it, kind of like uses a magnifying glass on it, so that you're able to work with something a little bit more pure and concentrated. One thing that we didn't do in lab this semester was hybridization or any of the blotting techniques. So probes are labeled short DNA fragments with dyes, usually some kind of fluorescent dye. Blotting is then taking those fragments from a gel and transferring them to a nylon membrane. In the case of DNA, that would be southern blotting, and RNA, that would be northern blotting. With molecular cloning, we can reproduce desired fragments of a genome. So we can use plasmids from prokaryotes as DNA folders and insert foreign DNA, which would be a fragment of the human genome. Once it's transferred to something else, like a bacterium, it would be called a transgene. And the host DNA would be the bacterium's DNA. So we're putting foreign DNA, fragment of the human genome, into one of these plasmids. Once those plasmids have that foreign DNA, they're called recombinant DNA molecules. These are created artificially, they're not found in nature, and they can express recombinant proteins as well. With reproductive cloning, this is a clone or identical copy of an entire multicellular organism. One example of this is Parthenogenesis, where an embryo grows and develops without egg fertilization. In some organisms, they lay eggs. If the egg is fertilized, then they become diploid and female, but if they're not fertilized, they remain haploid and become male. In some organisms, they can just undergo parthenogenesis and they strictly reproduce without egg fertilization. This is a form of asexual reproduction. And this has been shown in mammals as well. The first cloned animal that we did was Dolly the sheep in 1996. I'm sure you guys heard about that. And since then, we've cloned so many other animals, so many other mammals, and even some stem cells from embryos, which we use in therapeutic purposes. Genetic engineering is the alteration of an organism's genotype using recombinant DNA for the purpose of creating desirable traits. We see this a lot with a lot of the plants that we consume. Most of the plants that we consume nowadays have been genetically modified, especially things like bananas and watermelon. This is what a banana used to look like. All of these sections of like dark brownish material, those are all seeds. Our bananas nowadays don't have seeds. And notice that watermelon used to not have a lot of flesh on the inside. So they genetically modified them for more flesh so that they're more pleasurable to eat. But one of the main controversies in biology right now is genetic engineering in humans and using that to create desirable genotypes, such as parents choosing what color eyes they want their kid to have and so on. So that's a huge controversy right now. What we should genetically modify, what advantages and disadvantages there are to genetically modifying both humans and other species and organisms that we consume. Gene targeting is the use of recombinant DNA vectors to alter a gene's expression. So we can mutate or delete some genes to divulge gene function. Usually we do this in mice or other test animals. And this can help us learn what a gene's function is if we have not yet discovered that. Genetic diagnosis is testing for any genetic defects, especially if you know you have a family history of a certain genetic disease. Gene therapy is the genetic engineering technique used to cure disease. So it's the introduction of a good gene to aid in the cure of disease. They do gene therapy a lot with individuals with SCID or severe combined immunodeficiency, where they essentially don't have an immune system. So gene therapy is really one of the only resources that they have to try to treat this disease. Vaccines are typically produced with the use of a weakened or inactive form of microorganisms to cause immune response. They usually do pretty extensive trials of vaccines over long periods of time to test for any long-term effects. But luckily, if any vaccines are sped through the process, for example, in the case of the COVID vaccine, a lot of the severe effects of the vaccine, any severe adverse effects, would be noticed in the first couple of months, and luckily we've been able to test for that long to be able to determine whether or not there would be any adverse effects that were serious. So this will cause a weak immune response in the individual, sometimes causing what appears to be a weakened form of whatever they're being vaccinated for. As far as antibiotics are concerned, microorganisms naturally create antibiotics, especially fungi, in order to gain an advantage of them in their own biome. And then hormones, there are a lot of different ways that these can be manufactured. They can be extracted from other organisms. For example, sometimes we use pig insulin instead of growing insulin through microorganisms or cloning it. But it can be grown in bacteria or cloned from human insulin. So transgenic organisms are essentially genetically modified organisms. They are individuals that are modified to express recombinant DNA. In animals, these are used for studying mutation effects in humans, and in plants, these are used to create desirable effects, like we talked about in the banana and watermelon. But there are so many other examples. Genomes can be used to predict disease risk. I'm sure a lot of you have seen that 23andMe and things like that, to sequence your genome to figure out if you're at risk for developing any genetic diseases. Most of the common chronic diseases like heart disease are polygenic, meaning that several genes contribute to the formation of this disease. So it's not just one gene that's mutated that causes this. So when sequencing the whole genome, you're able to look at all of those different genes to determine what the risk is of actually obtaining those diseases. Pharmacogenomics and toxicogenomics, they evaluate the drug effectiveness and safety based on an individual's genome sequence. So They can do this through animals before they give a new drug to humans to see if there's any mutations that occur or any adverse effects that might occur. Metagenomics is the study of collective genomics of multiple species that grow and interact with one another in the same biome. This can be used to identify new species rapidly. And lastly, we've been looking a lot recently at microbial genomics. Microbes are actually a largely untapped field of energy. They could be used for biofuels, especially from algae and cyanobacteria. So this is an area in which there's a lot more exploration to be done. It would be great if we could come up with a new way of harnessing nature for energy resources rather than polluting nature for resources.